All right, so here we go. Introduction to Mendelian Patterns of Inheritance. This is the Chapter 11 lecture outline. A lot going on in this chapter. A lot of it we did hear of before, but there will be some new things added in here, especially some additional mathematical problems that will be introduced to in this problem set. So basically what we're going to do is look at a little bit of who Gregor Mendel is, look at Mendel's laws, look at Mendel's patterns of inheritance as associated with some human diseases, and then we're going to go beyond the scope of Mendelian genetics and talk about inheritance that is a little bit more complex than what we see in a typical mono and dihybrid cross, and you have like those one or two genes. More things will be playing a role there. So to start us off, we have this genetic disorder called phenylketonuria, also known as PKU. It's a human genetic disorder. We do know that phenylalanine is an amino acid found in many foods. If you do have PKU, you do need to look at the ingredients in there. Phenylketonurics are people who lack the enzyme that will ultimately break down this amino acid. What happens is excess phenylalanine will accumulate in their bodies and ends up causing nervous system disorders. Phenylketonuria is a recessively inherited trait, which means that people have to inherit 2 copies of the gene to have the disorder. Food labels inform phenylketonurics which foods they should avoid. So with that introduction, we get into a little bit about Gregor Mendel. And originally there was this concept of blending inheritance, where parents of contrasting appearance produce offspring of intermediate appearance. Over time, variation would decrease as individuals became more alike in their traits. Blending was a popular concept during Mendel's time, but Mendel's findings were in contrast with this. So we no longer view it as this blending inheritance concept, but we see it as more of a particulate theory. And he formulated this particulate theory of inheritance after his work with pea plants. And today we know that that particle that he speaks of in his literature is what we refer to today as a gene. Mendel also proposed the laws of segregation and independent assortment. And we'll look at those 2 laws as they apply to both genetics and our previous chapter, meiosis and reproduction. So inheritance involves the reshuffling of those genes from one generation to the next. Gregor Mendel himself, he was an Austrian monk. He went off to study science and math. He did focus in botany, studying plants, and math. The new math at that time was statistics. So he took that class at the University of Vienna and learned statistics and botany, which ultimately paid off in the end with his experiments in both using pea plants and doing genetics. So he conducted the breeding experiments with the garden pea, Pisum sativum, and he carefully gathered and documented mathematical data from his experiments. So there was the scientific evidence in the data quantitatively and also qualitatively that really supported all these facts that he was finding over time using pea plants. So he formulated fundamental laws of heredity in the early 1860s. Had no knowledge of cells or chromosomes as he was doing this. He did not have a microscope to work with, and experiments on the inheritance of simple traits in garden peas ended up disproving that blending hypothesis belief that once existed. So here you can see a picture of the pea plant and Gregor Mendel himself. Mendel did work with the garden pea, ultimately because the garden pea as an organism was a very good choice for genetics. One, it was easy to cultivate. It had a short generation time, so you could produce a lot of offspring in a rather short period of time. Normally, it could be self-pollinating, but you can control uh, cross-pollination by hand. Pollen was transferred from the male anther of one plant to the female stigma of another plant using a simple fine-haired paintbrush. You have both true breeding varieties that are available, and you could study simple objective traits, and we'll see some of those traits a little bit later on. So if you look just a little bit about flower anatomy, peas, like all flowering plants, are angiosperms, and in there we do have the stamen. The stamen is the male component of the plant made up of the anther, where the pollen granules are, that is situated on top of a filament. You also have the carpel, and the carpel includes the stigma at top, and the stigma usually contains like a sticky substance to trap the pollen granules. You have the style, and then you have the ovules inside the ovary there. So here you can see basically how Mendel did his work with the various traits of peas, studying the various traits of peas. So Mendel's laws, Mendel performed crossbreeding experiments. He used true breeding plants, and when we talk about true breeding, these are homozygous plants. He chose varieties that differed in only one trait, and because they differed only in one trait, we would call that a monohybrid cross, or a one gene cross. He performed reciprocal crosses from one generation to the next. And when looking at genetics, we know that the parental generation is always labeled P. Then you have the first generation, or the first filial generation, which filial is basically the Latin word for sons and daughters. Those we call our F1s, and the second filial generation, or offspring, are what we call our F2s, and so on and so forth. From this, we formulated the law. He formulated the law, not we. He formulated the law of segregation. The monohybrid cross done by Mendel, if you look here, we could see a true breeding tall plant, homozygous, for the gene of tallness, because it has two capital T's. And just a quick note there, if you recall from basic genetics, we use the letter of the dominant allele to represent the alleles for that particular gene. So here we're looking at the gene for P height, and we have two alleles, capital T for tallness and a lowercase t for shortness. So we are crossing here a homozygous tall plant with a homozygous recessive short plant. Both are true breeding in their respective trait. That would be our P generation. There are the gametes that we produce, and our F1s ultimately would become all heterozygous. And here they would be ended up being tall plants. But if we look again, crossing F1s, big T, little t, big T, little t, we get big T, we get a big T, little t, big T, little t, little t, little t. So what we get now is a 3-to-1 ratio, so 3 tall and 1 short. So you have a 75% chance there of having a tall plant and a 25% chance of having a short plant. And just a quick denotation here that you can see, the female gametes do get placed on the top of the Punnett square. So here we can see the eggs, and down here on the side are the male gametes, which represent the sperm. And remember that each little component here, or box, would represent one gamete. Therefore, you can only put one allele from each gene in that area. If we had less than one allele, or if we had more than one allele from each gene in the same box here, represented by a gamete, then if we go back to meiosis, that would have been non-disjunction occurring there. So Mendel's law of segregation, ultimately, here's what it is summarized. Each individual has a pair of factors, which we call alleles, for a trait. So 2 factors, or 2 alleles for each gene. And the factors or alleles are going to segregate or separate during gamete formation. So the formation of sperm and egg, that process there is meiosis. Each gamete contains only one factor or allele from each pair of factors. So that ensures that each gamete, whether it be sperm or egg, gets one copy of the gene. Hence, that's why we have haploid. Our gametes are haploid. Fertilization then gives the offspring two factors for each trait. The results of the monohybrid cross, all F1 plants were tall, disproved that blending hypothesis, but we did see that we did have shortness come up in that F2 generation. So Mendel's law is what we consider classical genetics. Each trait in a pea plant is controlled by two alleles, which alleles themselves are alternate forms of a gene. And we do have the dominant allele represented by the capital letter, which masks the expression of a recessive allele, which will be a lowercase letter. Principle of dominance there. Alleles occur on homologous pairs of chromosomes at a particular gene locus. So the gene locus is where that particular gene occurs, and where that gene occurs, of course, is where you would have the alleles. So ultimately, homozygous identical alleles, so that would be two dominant alleles or two recessive alleles, or you could have a heterozygous gene, which would be different alleles, one dominant allele and one recessive allele. So we know that homologous chromosomes, there is the gene locus. Homologous chromosome have alleles for the same gene at specific loci. And here you can see that upon replication, you get the chromosomes that will replicate, forming the sister chromatids. The sister chromatids of duplicated chromosomes have the same alleles for each gene. So these are the various traits that Mendel looked at in his particular pea studies. Here is the gene itself, so you could talk about stem length, pod shape, seed color, flower position, axial versus terminal, purple versus white for flower color, or pod color, green versus yellow. You could also see the different characteristics, that of the dominant and that of the recessive. So Mendel's laws basically have two different terms here. Genotype, which you should be well aware of. It refers to the two alleles an individual has for a specific trait. So basically what you're talking about here is the genetic makeup of the gene. What do those alleles look like? Are they both dominant? Are they both recessive? If so, that would be homozygous. Or do you have one dominant allele and one recessive allele? If that's the case, it would be heterozygous. You also have the phenotype, and the phenotype is going to refer to as the physical appearance of the individual. And the phenotype is all determined by the genotype of the gene. So the phenotype is expressed by the genetic makeup of that gene itself. Mendel's crosses viewed by modern genetics. The dominant and recessive alleles represent DNA sequences that code for proteins. The dominant alleles code for a protein associated with the normal gene function within the cell. The recessive alleles represent a loss of function. Ultimately, what we see then is during meiosis 1, the homologous chromosomes separate. The two alleles separate from each other, and the process of meiosis explains Mendel's law of segregation and why only one allele for each trait is in each gamete. Now, typically, what we would see there is that the alleles on the same chromosomes, especially if they are close to one another, or the genes, will move as a single unit. However, if you increase the map distance there, or the genes are found on other non-homologous chromosomes, what would happen there, or other chromosomes, the genes would basically separate from each other there. We'll look at that a little bit more in class. So we'll talk about map units, and we'll discuss that within the next few slides as we get into independent assortment. So Mendel's laws, we do have a two-gene cross, uses true breeding plants of differing in two traits, dihybrid. Mendel tracked each trait through two generations. It started with true breeding plants differing in two traits. The F1s showed both dominant characteristics. When the F1s were self-pollinated, he observed phenotypes among the F2s. From this, his results led him to formulate the law of independent assortment. Basically here, then, what is stated is the pair or factors for one trait are going to segregate independently of the factors for another trait. And this ultimately will allow for all possible combinations of factors to occur in the gametes. Again, the parental generation is the P generation in breeding plants. F1 is the first generation and F2 is the second generation. So if we look here we looked at plant height and pod color. So we have tallness and green pod crossed with a homozygous, short, homozygous yellow podded pea plant. And you can see that in the F1 generation, we have a heterozygous genotype for both genes. Phenotypically, it was a tall pea plant with green pods. But when you cross two F1s, you now see your variations of tall plants and short plants. Tall plants with green pods, tall plants with yellow pods, short plants with green pods, short plants with yellow pods. And you could go through that chart and see how it is all figured out. Ultimately, this then would be the 9:3:3:1 phenotypic ratio. So independent assortment and segregation during meiosis. Basically, the parent cell has two pairs of chromosomes that are homologous. The parent cell has two pairs of homologous chromosomes, which could either be there or there. So you could see the orientation of those homologous chromosome pairs. So all orientations of homologous chromosomes are possible at metaphase 1 in keeping with the law of independent assortment. Ultimately, at metaphase 2, each daughter cell has only one member of each homologous pair in keeping with the law of segregation. And you can see here that crossing over did not occur here. So all possible combinations of chromosomes and alleles occur in the gametes as suggested by Mendel's two laws. So here we can see the playing out of both of those laws, the law of segregation and the law of independent assortment. And from that, and we'll conclude with this for the first part of the lecture, with using Punnett squares. Punnett squares is a table listing of all possible genotypes resulting from a cross. So you get all possible sperm genotypes are lined up on one side, all possible egg genotypes are lined up on the other side, and then all possible zygote genotypes, zygotes are formed upon fertilization, are placed within the squares. And from that, we now use statistics, an area of statistics called probability. So Punnett squares use both the a multiplication rule and the addition rule for predicting the results of a Punnett square or a genetic cross. So Punnett squares allow us to easily calculate probability of genotypes and phenotypes among offspring. The Punnett square in the next slide shows a 50% or 1/2 chance. The chance of capital E, or the dominant allele, equals 1/2, or the chance of the lowercase e recessive allele is 1/2. So an offspring will inherit these possibilities. So if you get big E, it's 1/2 times 1/2, which is 1/4. Big E, little e equals 1/2 times 1/2, which equals 1/4. Little e, big E, 1/2, 1/2 equals 1/4. Little e, 1/2, 1/2 equals 1/4. So that's the rules for multiplication. You can then add to this and apply the sum rule, and the sum rule for probability, or the rule of addition, is going to allow us to add the genotypes that produce the identical phenotype to find out the chance of a particular phenotype occurring in that population from a genetic cross. So here you can see the Punnett square that we illustrated before. So you had that half chance for each of those alleles being passed on there to allow for these combinations down below related to attached or unattached earlobes. Mendel also was able to use test crosses to predict the genotypes of the parents. Individuals with recessive phenotypes always have a homozygous recessive genotype. However, individuals with a dominant phenotype have indeterminate genotypes. So it could be homozygous dominant or it could be heterozygous. And this is where a test cross would come into play. And we've done these a little bit back in sophomore year. So a test cross determines the genotype of an individual having the dominant phenotype. So if we look here, we do have one trait test cross. We cross a heterozygous tall plant with a homozygous recessive short plant. And ultimately what we see is that 1-to-1 ratio there. Or if you take the homozygous tall plant and cross it with a homozygous recessive plant, what would happen is you get all tall plants. So the difference is the F1 result from that cross a 50-50 as far as tallness and shortness, or is it 100% tall? And from that, you could predict your parental genotypes. Remember that always, in each situation, you're going to cross the one parent, the unknown parent, with the known parent's genotype being homozygous recessive. So we have an unknown genotype here and the unknown genotype here, and both of them are being crossed with that homozygous recessive to see whether it would be this genotype or this genotype for the unknown parent. So two trait crosses, an individual with both dominant phenotypes is crossed with an individual with both recessive phenotypes. If the individual with the dominant phenotypes is heterozygous for both traits, the expected phenotypic ratio would be a 1-1-1-1. However, we do see if they are heterozygous for both traits. Oh, no, wait, that's for a test cross. So I think we'll end there for today for this lecture, and then we'll pick it up with looking at genetic disorders, going a little bit beyond classical genetics, and doing some pedigree analysis. Thank you, AP Biology students. I hope you enjoyed this lecture. We'll do some practice in class. Have a good day.